Welcome back. And we are on our last leg of our swine technical session. Our next speaker would be also a pig practitioner. He is a manager in Pig Tech Veterinary Services. He's the manager owner of Depot Genetics Center from 2014 to present. He's the technical consultant of CJ Philippines Incorporated from 2020 to 2021. He's the manager, owner of Depot Farm Supply and Breeding Center from 2003 to 2019. He was a social person, a Purina salesman, Cargill, Philippines Incorporated from 2001 to 2003. Seaver Award for Entrepreneurship from CLSU Alumni Association. Central Luzon State University, Science City, Muñoz, Nueva Ecija, received August 2020. Colleagues, let us warmly accept Dr. Romulo M. Parayao with the title An Investigation on the African Swine Fever Transmission Potential of Common Housefly, Musca domestica, and its implication on biosecurity programs.
Fellow veterinarians, swine practitioners, stakeholders, guests, ladies and gentlemen, have a pleasant day to everyone. First, I would like to thank the Philippine Veterinary Medical Association for giving me the opportunity to share to you my experience on African swine fever outbreak. Also, I would like to thank the moderator for the kind introduction. Yes, I am Romulo Parayao, a farmer and a veterinarian. I have two swine facilities. One is the gilt multiplier, and the other is a boar station. Both were accredited by the Bureau of Animal Industry as a source of breeders for our hog farmers. This presentation, an investigation on the ASF transmission potential of common housefly, the Musca domestica, and its implication on biosecurity programs were product of curiosity. There are things that bothers me even until now. Some of them were still unanswered. First, why commercial farms with strict gate biosecurity measures and were hit by African swine fever outbreak? Second, why the spread of outbreak in one location usually started in one facility, followed mostly by adjacent facilities? The ASF was reported on July 2019 and officially declared by no less than our DA Secretary on September upon confirmation of the accredited OIE laboratory that 14 out of 21 samples submitted were positive on viral DNA of African swine fever. 6 months after the outbreak, the Central Luzon was already been under the red zone. This slide is a copy from the map of the zoning on February 2020. This is the current status of the outbreak in the country, taken last October 2021. The entire Luzon is already infected, and there are many islands in the Visayas, and a major portion of the Mindanao were also under the outbreak zone. The third question, if a fly can carry a whole bacteria and transfer it from infected facility to another, why it cannot carry an ASF virus? That's a big question. Is there really a chance that ASF virus can be transmitted by a fly? After this presentation, your guess will be as good as mine. Let's talk about the fly transmission potential to carry pathogens. According to the pig site, airborne and animal transmission of disease in pig herds, there are some viral agents that can be transmitted by housefly. the porcine reproductive and respiratory syndrome, and the porcine epidemic diarrhea virus. And there are other agents that can be transmitted by housefly. The bacteria Streptococcus suis, the Escherichia coli, Bacillus anthracis, and some species of Clostridium. The flies get infected from contaminated secretions and excretion from sick and dead animals. The fly can travel 2 to 3 kilometers and they are attracted to smell. The organism remained viable in the fly for up to 5 days, or depending on the livability of the pathogens in the environment. Let's compare the size of the pathogen that can be transmitted by housefly to African swine fever virus. The Streptococcus suis is 0.5 micrometer to 2 micrometer in diameter. The Escherichia coli is 2 micrometer long and 0.2 to 1 micrometer in diameter. The Bacillus anthracis is 1 to 1.2 micrometer in width and 3 to 5 micrometer in length. The Clostridium is 0.3 to 2 micrometer in width and 1.5 to 20 micrometer in length. The PRRS virus is 0.074 micrometer in diameter, and the PED virus is 0.19 micrometer in diameter, while the ASF virus is 0.2 micrometer average diameter. Comparing ASF to the Escherichia coli, ASF is simply about 10% of the size. Now, can it be added to the 100 pathogens that can be transmitted to housefly? We will find the answer at the end of the presentation. Let's talk about the ASF virus stability on the environment. The stability of the virus in the environment play a major role in its transmission. The longer it stays in the environment, the higher the chance that it can be transmitted to other hog facilities by fomites, by human, or by mechanical vector. The ASF virus is a large double-stranded DNA that belongs to the family Aspergillidae and genus Aspervirus. It causes contagious, hemorrhagic, and lethal viral disease of domestic and wild pigs, while warthogs are known to be resistant. The competent vector is the Ornithodorus ticks, where the ASF can still multiply inside the tick, waiting for the next susceptible host. And there are other known transmission: direct contact through body secretions, urine, and feces, contaminated meat and meat products that may end up in our pigs through swill feeding, fomites, as in vehicle, boots, clothing, and farm tools and equipment that we use in our everyday activity in the farm. There were also reports that some pigs are also contaminated with ASF and some underground water. It has been believed that mechanical transmission of stray animals and rodents are accepted paths. But stray animals and rodents will stay in one location only, where food is abundant, and they will not travel 500 meters from their location. It is very important that we identify a potential mechanical vector of ASF, as it plays an important role in the spread of disease, and it will have an impact in our eradication program. According to Vizcaino, the vector is the most difficult to eradicate. Every seminar that we attended about ASF, there is this kind of slide, the ASF survival time in the environment. For this particular presentation, I used the FAO slide. The virus in an uncleaned farm left after the removal of sick animal may still be infective for a period of 11 days in feces, and up to 30 days in undisinfected pig pens. Fly that may swarm the area can transfer the virus to the neighboring facilities. Leaving these contaminated facilities uncleaned and undisinfected extends its life of infectivity. And after the removal of figs, they provide millions of viral particles available for mechanical transmission by human or the flies. Let's talk about the fly. The fly is a perfect sample of a disease transmitter. They have wings. They can travel 2 to 3 kilometers between facilities unnoticed. They have 6 feet and a proboscis and can have a direct contact to the contaminated area or to the pathogens itself. In fact, most of us believe that fly can carry pathogens, but none of us include fly control in our biosecurity measures. The anatomy of the foot of housefly. They have 2 foot pads. They have adhesive property with tiny hairs and a gripping claw. Structurally, the housefly is suited for picking up pathogens. The continuous movement of flies from filthy substances like human feces, animal excreta, dead carcasses, garbage to our food or feeds make them perfect candidate for a disease transmission. The life cycle of a housefly. An adult female can lay up to 120 eggs and 5 times in her life. The egg hatch in an hour to form the larva or the maggots. Eventually, it migrates into a suitable location and forms to the pupa. Depending on the environment, the pupa can develop into adult fly in 2 to 10 days. The fly is important in degradation of organic waste. No contamination, no problem. Before the ASF came into the Philippines, there was no problem about the fly. The question is, if the ASF virus detected in the fly is still infective. The lifespan of a fly is 15 to 30 days. If they catch the virus may still be infective even after it dies. There are over 100 pathogens associated with housefly. The housefly play no important role in the life cycle of these pathogens. They simply act as a mechanical vector. Now that the virus is present in our environment, it can be added to more than 100 pathogens that can be transmitted by a housefly. Imagine this dead pig. If this died due to ASF, the flies that may have contact with it will definitely be contaminated with ASF virus. This may be a sample of pigs that transported by the farmer into the mountain or thrown into the riverbanks, not knowing the danger that it brings to other hog raisers. Uncovered mortality pits may also be a source of contamination. How does flies transmit pathogen? They transmit the pathogen through their bodies, legs, mouth parts, sometimes through its vomitus and feces. The infective dose of each pathogen varies greatly. Sometimes just a few required to cause a serious infection. Again, the fly must be contaminated first before they can spread the disease. It does not need to bite like a horsefly or tsetse fly in order to inject the pathogens. The ASF virus is transmitted orally. The fly that left the virus on the wet floor, on the feeding trough, and the nipple drinker can be ingested by the pigs. As Sandra Blom says, 1 drop of blood from dead pigs due to ASF contains millions of ASF particles, and 1 drop can initiate an outbreak. This investigation was done like any ordinary way of determining suspect of a potential disease occurrence. The presenter is not a researcher, but a veterinarian. There is no specific basis for this methodology. We simply catch flies, send it to laboratory for testing on the presence of viral DNA. We have many friends in the industry who trusted us, and they are open to share their experience, including the ASF outbreak. The Farm A is a 4,000 head commercial farm in Baliwag, Bulacan, reported an increasing number of baby mortalities associated with fever. We reported it to the Regional Field Office of Region 3. They collect samples, and upon confirmation for the presence of ASF in the area, they assisted them in the population of the facilities. The Farm B is a backyard racers in Caldao, Pampanga. The cases start with off-feeding of gestating sows followed by abortion. And there was a reported mortality in the following days. This also was reported to the DA Region 3, and they assisted them in the population. These two hog facilities with initial report of high mortalities associated with high fever and abortion were selected as participants for this investigation. Verbally, we asked them to support in the collection of lice for testing of possible ASF contamination. We agreed to collect 100 lice, and this will be submitted for real-time polymerase chain reaction test. The Farm C is a 500 capacity breeder farm in Campus Talavera, Nueva Ecija. It is situated 863 meters from a recently depopulated infected premises on January 7, 2020. The barangay celebrated their fiesta on December 13, 2019, and there are rumors that they buy carcasses from kolong-kolong mobile vendors. Before the New Year of 2019, there were reported cases of mortalities in the said barangay. The first reported case in this farm of high fever on gestating sow is on January 15. That leads to abortion the following day. There are also 4 gestating sow that did not eat their morning meals on January 16. This facility was reported to Provincial Veterinary Office of Nueva Ecija for possible scheduling of the population. January 18, when the PBO Nueva Ecija depopulate the area, and we agreed to retain 20 apparently healthy piglets to observe if it is true that once the farm gets infected, all pigs may die. From January 18 to February 10, all the remaining 20 piglets died due to ASF, and the last pig was used in this investigation. The sample was collected on February 10 on Farm C. It's hard to collect 100 flies at a time. So we covered the dead pigs with mosquito net, and the caretaker catch fly using the fly net and put them inside the mosquito net. Let them stay there for overnight. Upon confirmation on the availability of reagents on Blitzkrieg Laboratory in Central Luzon State University in Muñoz, Nueva Ecija, we collect the live flies and submitted a sample to the laboratory. The laboratory technician was advised to dissect and do a separate test for legs, body, and the whole fly to identify which part may have the virus. We requested 3 runs for reverse transcriptase polymerase chain reaction test and 3 runs for ASF nanogold biosensor test. Farm A, with 4,000 head capacity, they simply catch fly everywhere. And the sample were macerated, the whole fly, and they ran 3 RT-PCR. The sample were submitted on October 23. For Farm B, from backyard racer, the same. They catch fly everywhere using fly catcher. The same procedure, macerated whole fly, and run only 1 RT-PCR test. Both of them were submitted on October 23 at the Regional Animal Diagnostic Laboratory in San Fernando, Pampanga. For the Farm C, 500 head capacity breeder farm, we enclosed the dead pig with mosquito net and put the caught fly inside for 12 hours. And then we collect the live flies, and submit it for sample. We ordered the laboratory to have a different test for legs, body, and the whole fly. And they ran one each test. The sample was submitted on February 10. The results showed that Farm A and Farm B both submitted to Regional Animal Diagnostic Laboratory in Region 3. Viral DNA is not detected. For Farm C, for the reverse transcriptase polymerase chain reaction test, they detected positive on legs, negative on body, and negative on whole fly. For the Nanogold biosensor test, the result shows highly positive on legs, negative on the body, and positive on the whole fly. This investigation showed that ASF virus transmission potential of housefly as a mechanical vector is possible. Erms et al. In 2020 in Estonia also stated that flying insect help in the mechanical transmission of virus. The viral DNA was also detected in Lithuania by Tursinabicin et al. After testing Musidae, Caliporidae, and Tabanidae. Similarly, they believe that these fly play a role in mechanical transmission of ASF virus. It was observed that a series of outbreaks are usually adjacent to each other, despite of culling and removal of pigs in 500-meter radius. The current measures focuses only on removal of pigs. Cleaning and disinfection, which is part of the recommendation of the FAO, were not religiously being followed. Leaving the infected premises unclean extend its infectivity of the area, causing the possibility of mechanical transmission from exudates and fecal matter. 1 out of 3 fly samples collected from ASF infected farms tested positive for viral DNA. Therefore, it can be concluded that flies can mechanically transmit ASF virus on their legs and can help in the spread of infection in the nearby facilities. Now we can add the ASF to the over 100 pathogens that can be transmitted by housefly. The implication on our biosecurity measures. Effective biosecurity should not only be designed to protect the pathogens from coming in, but also prevent them from going out. Upon the release of the result of this laboratory test, the presenter posted it on his Facebook account. A continuing study on the possibility of housefly as a transport carrier of African swine fever virus. Today, results showed that they are potential carrier, and therefore we need to establish a fly control program and add it into our existing biosecurity protocol. The result will be further presented in a technical paper, but due to the necessity with the current ASF outbreak, we need to have it as public for our immediate action. This may be too late in some regions, but it will also be good for future and repopulation program of our industry. The results were also forwarded to our BAI director, then Dr. Ronnie Romingo, and he replied, "Congratulations, you did something to improve our knowledge on ASF transmission." Also, a colleague posted on his Facebook page, GeneStar Swine Technologies, a PCR test of housefly facilitated by our local swine expert showed that housefly can mechanically transport the ASF virus. The amount of filth in the legs of the fly may carry millions of viral particles. The legs are the one directly in contact with infected material. The test result stresses the importance of undertaking serious fly control on our farms in an effort to prevent further spread of ASF. Also, a colleague, Dr. Hill Austria, posted in his Facebook account, swine island, the only connection to the outside world is the air. No flies, no rats, no birds. In his opinion, fly play a big role in the spread of ASF within a kilometer. He posted it on October 29, 2020. Also, Dr. Pulprem Pariwat of Thailand shared a picture of a farm covered with nets. Self-defense, especially on farmhouse, often farm. It is very important, especially birds and flies. Let's find a way to protect yourself. When I saw the post, I introduced myself to Dr. Pariwat and asked the question, may I ask if we have basis for this measure? Is fly considered as vector of ASF in your country? Thank you for your response, and I hope to collaborate with you about ASF. And according to him, I saw someone in the Philippines post on his Facebook, and my student in Vietnam also found flies carry ASF. A backyard research in Region 2 made his testimony. Totoo po yan, Doc. Living testimony po ako. Kapitbahay ko po, wash out na. Yung mga naiwan po sa piggery ko na 20 heads ay standing pa rin po at healthy. Also, a physician from Bulacan. Ako po, isa na lang ang natitira. Buti patining ko hindi tinamaan. Nilagyan ko kasi ng kulambo yung mga kulungan nila. To encourage discussion, tinanong ko po siya, para saan po ang kulambo? Nakita ko po, kasi Doc, dati yung post mo na positive sa ASF, yung mga langaw na pinalaboratory mo. Kaya nilagyan ko ng kulambo yung patining ko. Ayun, nakasurvive naman. Salamat pala sa post mo, Doc. Kung hindi ko nabasa, baka lahat ng alaga ko naubos. Our recommendation for the government. Research institutions should fund more research studies among common disease vectors, like flies and other insects, not only for ASF, but for other important diseases. And also to include immediate cleaning and disinfection of facilities after removal of pigs in infected premises. This is a picture of dead pigs with flies. And also this is more flies. For our hog raisers, we have to include fly control in our biosecurity measures, either chemical or physical program. According to Dr. Vizcaino, an outdoor production was the last area to get rid of the ASF virus. One of our recommendations is to make a farm covered with nets. Farm netting is the new normal. Our farm should be a no-fly zone. This proposed building design was posted on my Facebook, and this was designed to prevent not only the flies, but also other stray animals. If we cannot go tunnel ventilated for backyard raiser, we should try to enclose our farm with nets. My take-home messages. Our biosecurity program will only be effective within our farm premises. The government should implement a more strict animal movement control to prevent the continuing contamination of our environment, the roads, the slaughterhouses, and our markets from the newly infected zone. Thank you very much for listening. And if you have any questions, I will answer them at the Q&A portion.
Thank you very much, Dr. Parayaw, for selflessly sharing your knowledge with us this evening.
Hey, for you, babe.
Sus! Gutom lang yan. Sugod na sa Hey Hey! Or call Hey Hey Delivery Hotline 8687-1010. Pwede rin online sa heyheydelivery.com at sa Foodpanda. We are now accepting GCash, credit, and debit card payments. Hey Hey, ang napakasarap na sagot sa tawag ng gutom.
We're in the business of combining ingredients and expertise, solutions and passion, curiosity and the unknown. Sometimes the results are expected, but sometimes they are extraordinary. The new IFF is an extraordinary combination, a company and team unlike any other. We fuse diverse perspectives and skills, starting a chain reaction that generates better, not just more. We strive to be essential, fueling our partners with what they need today, while inventing the foundations of what's next. This aspiration comes with a responsibility to do better, to challenge the status quo, to help people live healthier, more vibrant lives, to protect and extend the resources of our planet. The new IFF is extraordinary because here, where science and creativity meet, there is no limit to what we can solve for.
For our last Q&A of the evening and of the breakout sessions of this year's PVMA Convention, let's bring back Dr. Santiago and Dr. Romulo Parayaw. Hello again, doctors.
Good evening. Doc
Good,
Romy.
good evening.
Good evening.
Good evening sa lahat.
Ako po'y humahanga sa inyong ginawang study. No? May mga questions, may mga katanungan po tayo dito para mabigyan linaw po. Ano po? Si Dr. Alice Bungay, I agree that flies are efficient mechanical vectors. Just want to clarify that by your statement about the transformation of virus.
Ma'am, I think wala naman akong nabanggit na yung virus ay kayang mag-transform. Actually, hindi naman siya nag-mutate, no? Wala naman yata akong nabanggit na nag-transform siya. No? Saan part po kaya niya na
Alright.
na ano?
Baka lang po hindi na nag-lag or whatever.
Mhm.
Doc, my question is a little bit on the practical side kasi kung cost-benefit analysis ang titignan natin. Ang paggamit po ng pesticide is mahal. So, ang paggamit po naman ng net ay mura kumpara sa paggamit ng pesticide na tuloy-tuloy. Gaano po ka-efficient ang pag-transmit ng ASF virus ng langaw? Gaano po siya ka-efficient? Ibig sabihin po ba may ilang langaw po ang kailangan yung na-quantify po natin? Ilang langaw po ang kailangan para masabi natin na ops, hindi to no-fly zone nga tayo. Pero ang langaw ho naniniwala ho ako doon sa sinabi niyo, lumilipad na sakit ho 'yan. Flying disease ho 'yan eh. So, ano po ang efficiency ng fly in terms of transmitting ASF?
Actually, Doc, this is an investigation is just the first preliminary kumbaga sa mga researchers. So hindi pa ito umaabot dun sa determine natin yung dose or yung number of antigens na nakasama sa fly. As the study, yung title is a transmission potential pa lang. When it comes dun sa number, I believe the virus is hindi parang antibiotic yan na pag binigay mo sa hayop ay i-degrade niya na and then tatapon na. When the virus enter the target host, it will multiply. So, a single na cells na mapasok niya, pwede niyang maparami yung virus doon. Actually, hindi naman niya agad pinapatay yung mononucleated cells na napasok niya. And yung virus ay napoproduce through budding. So, hindi niya kailangan na marami at the beginning, pero dadami siya dahil nandun na siya sa target host cell. So, probably kaya mayroong incubation period na 4 to 19 days. It depends on the number of viral particle na nakapasok ng una. And also, may research din tayo na binabanggit sa mga previous forum na kapag laging mayroong virus na pumapasok sa katawan, kahit paunti-unti, especially kung sa tubig dadaan, mas mabilis siyang ma-absorb. So probably, kung sa tubig, baka hindi nagre-release ng masyadong maraming hydrochloric acid ang ating sikmura. So hindi siya na de-deactivate. Similarly sa PIDS kasi mas kaunti yung nakikitang yung virus dun sa yung potential na yung virus ay mai-transmit sa PIDS kasi nadadigest yung PIDS kasama na probably yung virus ay naapektuhan din ng mga acids and enzymes sa stomach ng hayop. So, wala pa tayo dun sa level na yun. Actually, last year pa na PVMA, kinukumbinsin na ako na i-present ito at medyo kailangan ng industry. Pero hindi po ako pumayag dahil I believe kulang pa nga. Bali, 3 farms pa lang itong nakolektahan natin ng sample. Unfortunately, ano po ito sariling gastos ito alam niyo naman ang PCR mahal and tinamaan na nga yung farm natin. Wala na tayong source of income during that time. So hindi ko na na-push through, especially nung lumayo na yung outbreak sa Nueva Ecija. Napunta na ng Cagayan, napunta na ng malalayong lugar. So hindi ko na pwedeng sundan. I tried to reach out some LGU veterinarians conducting yung depopulation to please collect flies and then send sa akin. Pero hindi daw nila kayang gawin kasi medyo loaded na sila. Overwhelmed na sila ng trabaho during that time. So, yun, hindi pa siya ganun kadami ng samples na nagawa. Thank you.
Thank you, Doc. Thank you, Doc, for the clarification. My take ko kasi sa nangyari is that flies can be mechanical vectors. Now, ang sa akin po personally, I would like to know yung infective dose kung ang daan nila ay through flies. May mga documents and literature states that 1 to the 10th ang power of 10 ang infective dose kapag
Nasa
sa
liquid.
water or sa water type. Yes. Pagka nasa solid medium naman po, mga around 1:50. Power of 50. So, may mathematical na representation na nakita. I guess they have to eat around mga 50 to 100 flies ma-ingest so that the virus would be enough to be infective. The problem with that, ang isang trait po ng virus ng ASF is that continuous low-grade infection will become a full dose infection din. Kaya po mahalaga pong makita natin na yung mechanics on how the virus is effectively transmitted.
Yeah. Tama, Doc.
Okay,
Ano?
there's
Kasi
this
hindi naman siya, Doc, na de-degrade sa katawan once na pumasok. Nagmumultiply siya sa loob. So, hindi
Yes.
kamukha nung antibiotics yan na mayroong minimum dose na kailangan para
Yes.
mag-express.
Dr. Alice Bungay has a question. What about other insects like blue butterflies, green butterflies, or black butterflies, or stable flies? Is ASF transmission fly species dependent? Your take, Doc.
Sa palagay ko, pare-pareho sila ng anatomy ng yung anatomy ng kanilang legs, Pare-parehas lang 'yan. So, kung talagang sila ay kasi sa atin yung Musca domestica, pag may mga malapit na poultry, alam niyo naman 'yan, pag harvest season, napakarami niyan, no? So, ang daming open yung mga housing ng backyard racers ay open-sided, no? So, talagang conventional 'yan. Kung biglang dumami yung fly, every 30 days mag-harvest ang poultry. And then yung mga open-sided na mga backyard racers. Kaya sila naman yung madalas na tamaan yung mga open-sided. No? So hindi siya hindi siguro natin pwedeng sabihin na kumukonti yung dala ng isang langaw hindi na siya pwede. Paano kung batalyon ng langaw yung pumupunta doon sa farm? Nakikita natin 'yan. Pag pinasok yung farm natin during the harvest season ng mga poultry, by the thousands ang fly na pumupunta sa iyo. So, ganoon din, no? Kung yung ASF virus is available in the environment at siya yung nadaanan ng fly na yan, may tendency na magsimula yan. And once na may isang baboy na natinamaan sa loob ng farm, definitely it will spread inside na. So, hindi naman kinakailangan na yung buong piggery 10 agad ang tamaan. Kahit isa lang ang mapuruhan ng virus, it will cause the outbreak inside the farm.
Okay. Doc, if you do biosecurity audit, ang reality ngayon, pest control is not ranked that high, no? Based on your experience, Doc, ano ang implication or importance ng study na nakita nyo na as fly as potential mechanical vectors? Kasi academically, nobody can question your result because in itself, it is a data. So, ang akin lang pong tanong dito is, pag nirank ko natin siya compared sa other established na vector or not vector, transmitter of ASF that can be found inside the farm, how would we rank pest control and how would it impact yung ating biosecurity program or protocol?
Bali, Doc, yung tanong mo parang babase muna ako dun sa mga kwento na ng mga naglagay ng nets. Si Doc Hill Austria, nag-ask ako ng permission i-share yung kanyang post sa Facebook. And yung farm nila, malaki yon, commercial farm yung, breeder farm. And ang findings nila, napaliligiran na sila ng infected na mga backyard racers. Hindi napasok yung farm nila. And also, meron din akong mga friend, nakasalukuyan na nag-repopulate. And according to them, ang payo ng feed company na kausap nila na unang nag-conduct ng sentineling ng animals, part na ng protocol yung mag-enclose ng farm using nets. And yung mga hindi tinamaan, dito sa area namin, naka-nets na rin. So, yun nga, sabi ko nga po, kaya ipinost ko na yan agad even without the number of replicates para sa isang scientist na dapat maging conclusive yung research, kailangan marami kang number of replicates. No? Kaya lang, sabi ko nga, kung makakasagip ako ng isang magbababoy because of this result, success na ako doon. Kumbaga, satisfied na ako. No? So marami tayong client na follower natin sa Facebook na nag-enclose ng kanilang net, ng kanilang piggery. And they, yung baboy nila ay naging protected, hindi tinamaan. So probably siguro isa na rin 'yon sa gusto natin ngayon sa mga biosecurity protocol natin. May color coding tayo sa farm. Merong black, meron tayong red or kung ano man yung ating segmentation dun sa loob ng farm. Siguro pagka kasi meron ng net, wina-warningan ka na noon na bago ka pumasok diyan dapat ay nakapagsabon ka na ng kamay, nakapagpalit ka na ng tsinelas, ng damit, no? Yung ginagawa nating color coding ay more on psychological, hindi mo nakikita. Pwede kang mamali. Pero kapag naka-enclose na yung farm mo, yung babuyan mo, automatic bago ka pumasok diyan, dadaan ka sa pinto. So makikita mo na yung disinfectant, makikita mo yung bota. Meron ka ng physical barrier bago ka makapasok. And I think yun na yung malaking tulong dun sa mga gumawa noong enclosement ng kanilang mga piggery. Mga backyard racers yan, pero they survive. So yun, mga testimonials po yun ng mga nag-enclose na ng kanilang facility. Mapa-backyard pa o mapa-malaki man. Kasi impractical nga sa atin mag-tunnel ventilated yung small farmers. Thank you, Doc.
Very good insight yan, Doc. And as I mentioned earlier, ASF is becoming a social disease. Kailangan talaga ng collaboration or malasakit ng bawat isa para hindi to kumalat ng ganun-ganun na lang. At lahat ng ating pwedeng magawa para maiwasan ang pagkalat nitong sakit na to ay kailangan nating gawin. Napaka-noble po ng inyong approach na yung backyard po ang isang yung study nyo ang magiging beneficiary nito ay backyard. Pag maliit po ang sow unit, mas madali po itong ma-contain o madali po itong i-manage. Alright. Doc, may question po dito. Ang tanong po nila is coming from Dr. Jose Alexander Abella. For how long will the ASF virus persist in the fly's body?
Ang basehan ko po nung aking kwento kanina is yung FAO na sinasabi na ang ASF kayang mag-survive sa environment up to 30 days sa undisinfected facility o yung iniwanan noong na-infect na ground zero. So kung yung lifespan ng isang langaw hanggang 15 to 30 days lang. Kung nakuha ng langaw yung virus sa nearly dying animal, yung naglabas pa lang ng dugo sa ilong, doon siya nadapo sa blood nung lumabas na yon. Ibig sabihin, sabay sila. Ibig sabihin, kung yung fly na yon ay nasa half of the life na, kung halimbawa 15 days old na yung fly na yan, dumapo siya doon sa freshly na blood, mamamatay na po yung langaw na yan. Pero yung virus, possible infective pa rin. Kasi 30 days siya sa environment. So, yun po yung yung naiisip ko on my side as a veterinarian. So, although wala pang nagsasabi o wala pang studies na namamatay na ba or deactivated na ba yung virus sa paa ng fly at the number of days. So, pero sa akin, pag pinagkumpara ko yung dalawa, yung buhay ng langaw versus yung buhay ng virus sa environment, mauuna pang mamatay yung langaw bago mamatay yung virus sa environment. Thank you, pa. No sounds, Doc. Mute.
Kami pong naging interesado doon sa ginawa niyong papel. Pag nakita niyo po yung messages natin, nakalagay ho dito sa wala ho sa Q&A.
Ay, wala ba?
Wala hong tanong. Ang mga suggestion po ang lumalabas. Example po nito ay galing kay Dr. Luis Luciano S. Gray. One thing that came across my mind was to use a piece of ASF negative tested meat and letting flies contaminate it, then test if the meat after. Is this possible to also prove the vector capability of flies? Kumbaga po, ang gagawin po niya, negative yung karne, padadapuan sa langaw ho na nag-swimming sa ASF positive na lugar at itetest ho natin yung karne. Okay. Ang take ko po dito ay number one, ang virus ho hindi naman kailangang bagay, kailangan po ma-demonstrate po siya sa tissue. Kaya lang, hindi ko ho alam kung magmumultiply sila kung patay yung host nila. So, your thoughts, Doc?
Malamang po hindi magmumultiply kasi pumapasok ang virus sa nucleated cells natin through phagocytosis. So, a dead animal, hindi naman magkakaroon ng white blood cell na hihigupin yung virus papasok. So, it will be as good as ano lang yan, na contaminated lang sa labas, hindi siya papasok. So, there will be no multiplication. Yun po yung parang unawa ko doon. Pero pwede po yung studies na yon, why not? Para lang malaman mo talaga kung magkakaroon
There you have it.
Kung magkakaroon ng virus doon sa karne. Probably ang susunod na papagawa ni Doc doon or ni Sir Luis ay ipapakain yung karne sa hayop, parang swill feeding.
Sa baboy.
Kung makaka-infect. Baka yun ang susunod doon sa mga gusto niyang mga curiosity din.
Yeah, oo, parang we will prove na the virus is still viable. No? If it's the DNA material na marirecover will be infective, not just fragments. Kasi ayun po ang problema kapag nagtetest ho tayo ng virus. Hindi po masasabi sa atin kung infective yung material na nakita natin, bagkus kailangan din ho natin i-consider yung source kung saan ho natin kinuha. Okay? There's this question from Dr. Giselle Guzman. Besides putting net, what other biosecurity improvement were done in concurrence to the installation of the nets? Perhaps additive improvements have contributed to the prevention of entry of ASF. Your thoughts, Doc?
Probably kasi yung mga tao aware na ngayon. Before kasi hindi naman sila aware yung biosecurity na yan. Wala namang kahit na tayo pagka nag-invite tayo ng magsiselect ng guilds before, pag pinagpapaligo mo, maliligo before mag-entry, magpapalit ng damit, parang ang tingin nila napaka-selan mo, no? Well, in fact, yun yung tama eh. So, hindi ko kung breeder farm ka lang, ikaw lang ang gagawa ng magpapa-shower in, shower out, change clothes, change footwear. Sa backyard racers, nung pumutok yung ASF, pumasok din sa isip nila yan. No? So nagsimula na rin silang maglagay ng boots. No? Nagsimula na rin silang mag-change ng damit and nag-disinfect na rin. No? Probably nung mag-net na sila, kasama na yung ibang biosecurity na basic, na magsabon ng kamay, ng paa, palit ng damit, no? Kung hindi mo kayang maligo. No? So, palagay ko kasama na rin yan. It will add up dun sa total biosecurity na ng backyard racer. Kasama siguro yung netting. Nababanggit natin yung netting. Parang nabanggit noong Vice Chair ng National Federation of Hog Farmers, si Sir Alfred Nang, na lahat ng kanilang sinesentinel na yung nagre-receive ng sentinel animal, pinapayo din nila maglagay na-i-enclose yung area sa net. No? So, malaki na ang nagtumatanggap doon sa possibility na yung fly is a potential mechanical vector. Nawala si moderator. May tanong po si Serapine Garciano habang hinihintay natin si moderator, si Doc. Good evening. Do you have any recommendation po in collecting samples of flies without any chance of contaminating the sample? Let's collect only the live flies. Huwag tayong kukuha ng mga fly na namatay na nasa flooring. Definitely, pati yung wings niyan o yung body niyan, pwede nang makontaminate. Pero kukuhanin natin yung buhay para sigurado tayong yung legs lang ang nag-contact sa flooring, para hindi makontaminate yung sample natin. So yun lang po yung ginawa namin. Kasi pag pupulutin mo yung mga patay na flies, magbibigay yun ng result na yung body ay most probably positive. Kasi yung virus nasa environment na talaga.
Good evening, good evening.
Yun, balik.
Nakabalik na. Forgive me for the disruption ng connection. Okay. We have a question, Doc. The question here is, do you have any recommendations in collecting sample of flies without the chance of contaminating?
Doc, sinagot ko na yun habang nawala ka.
It's
So,
a question
sabi ko na,
given by Dr. Serafin Jr. Garciano.
medyo pinakialaman ko, Doc, yung
Okay,
role mo.
okay.
Binasa ko na yung tanong. So, yun lang. Ang sabi ko kanina is let's collect
Go
the
ahead, Doc. Go ahead.
live flies lang. Para hindi yung patay na nasa flooring. Kasi gugulong na yun doon sa environment. There may be a possibility na makontaminate yung body. So only live flies lang. Thank you, Doc.
Okay. Good evening, Doc. Okay. Thank you, Doc. Good evening, Doc Parayao. Do you have any study if mosquito can also transmit the ASF virus to other pigs? From Dr. Helen Corpus Sibilia.
Sa ngayon po, Doc Helen, wala tayong nagawang pag-aaral.
I will repeat the question. Good evening, Doc Parayao. Do you have any study if a mosquito can also transmit the ASF virus to other pigs?
No. Doc, sa ngayon wala po kaming studies about the mosquito. No? Hindi ko siya na isama doon sa pwedeng ipatest.
Doc, nakuha niya? Nakuha po?
Aba. Nakuha po. Nawala ba ako?
Nakuha po.
Kuha. Okay ako, Doc. Naririnig po ba ako? Doc Amiel, buka yata narinig ako ng iba eh. So mukha pong yung ating moderator ang medyo nawawala ng signal. No? Balik tayo ulit. So habang hinihintay
Hello,
po
Doc.
natin.
I'm sorry. My connection is not at
Stable.
par ngayon.
Wala ulit, Doc. So may tanong po dito si Doc Francis Catalbas. No? Good evening po. Is it possible to treat the net with chemicals that kills the flies that also safe to people and the animal? Thanks. No? One of the reason why we recommend net, no, para po one time yung gastos. No? Hindi pa ulit-ulit. Kasi if we will use chemical, paulit-ulit po yun. No? So, kaya po natin ni-recommend yung net para minsan na lang.
All right. Doc, thank you very much for your selfless effort to contribute sa research and to fight against ASF. And as I have mentioned earlier, I believe as a farmer also na kakayanin natin bumangon mula sa sitwasyon na ito. With that,
Yes, Doc.
thank you
Thank
very
you.
much, Doc. With that, I would like to thank the participants. Thank you everyone for the question. Thank you very much for the interest, and good night to all.